Hi, I'm Gary Stix. I'm an editor at Scientific American, and I'm here with Robert Malenka, who is the Nancy Friend Pritzker Professor in Psychiatry and Behavioral Sciences at the Stanford University School of Medicine. And he's also the Deputy Director of the Stanford Neuroscience Institute. I should call him Rob. Rob's work is a major hub of research on synaptic plasticity, and in addition to all of the basic work that you do, your work also is focused on how that applies to real-world disorders like drug addiction, which is a major topical social issue these days, and the whole world is hankering for knowledge about the underlying processes so that it's possible to do something for people who are unable to kick drugs, whether it's nicotine, alcohol, cocaine, or amphetamine. So I'd like to start by talking about what your presentation is going to be about here at this conference. It really focuses on social reward and the fact of serotonin release in the nucleus accumbens promoting prosocial behavior. So could you tell me about what this study is doing, and also related to the larger picture of what you're doing in your lab? Because this is only kind of a component of everything you're doing in your lab, but it fits kind of like a piece in the puzzle in that larger piece, right?
Exactly right. It's a piece of a jigsaw puzzle.
Okay.
So I think it might help if I give you a little bit of a historical perspective about my work. As you mentioned, I started my career really focused on, trying to figure out the detailed molecular mechanisms underlying what we term as synaptic plasticity. And the assumption behind that work was very simple. I just reasoned that the bidirectional control of synaptic strength is the term, synaptic transmission, by activity, by changes in the patterns of activity, just intuitively must be a fundamental mechanism underlying the circuit modifications of many, many different forms of what I will term experience-dependent plasticity. So the big question is, you have an experience, whether it's sitting in a classroom, learning 2 + 2 = 4, whether it's a drug experience, taking cocaine or heroin, whether it's a stressful event that impacts your nervous system, changes patterns of activity, and depending on that experience, that experience modifies your memories, your future thoughts, your future feelings, and your behavioral response, just other kinds of stimuli. And the question is, how does that happen? So I made that assumption that I think turned out to be true, that it's just a
How far does that go back?
I mean, the work I started on synaptic plasticity mechanisms, I began, probably in the mid-'80s. So, and I continue to work on that, just really core molecular mechanisms. Again, with the reasoning that this is just a fundamental process that our brains use for all sorts of important phenomena, and important functions of the brain. That led me to start, that work was very basic in its topic, what I was trying to figure out, and I continue to do that sort of work. Mostly in collaboration with Tom Suhoff, who's a colleague at Stanford, whose lab is next to mine. But beginning in the late 80s, early 90s, I began to wonder, well, is this assumption true? Is it really true that powerful experiences modify synaptic transmission that has caused synaptic plasticity? And if so, how does that happen? Where does it happen? And how might that be actually clinically relevant? And the reason that was of great interest to me is I'm a psychiatrist. I actually did a psychiatry residency at Stanford. I haven't seen a patient other than my colleagues for about 25 years. But I've always had this interest in trying to figure out ways of applying my work to important clinical problems. So beginning in the 90s, early 90s, I started thinking, how could I give an animal a powerful experience and look for synaptic changes? First to see whether these plasticity mechanisms actually occur, and then to see if that might be important either pathophysiologically or perhaps even therapeutically. And that led me into the drug addiction field, because in the even in the early 90s, there were people talking about maybe drugs of abuse are usurping in a pathological way some of these core plasticity mechanisms in specific neural circuits in the brain, and specifically in what we call the mesolimbic reward dopamine circuitry. So as I'm sure you're aware, and I don't know if our listeners are aware, there is a highly evolutionarily conserved neural mechanism, what we call a circuit, that tells us when something feels good. And it's kind of intuitively obvious why that's important. We need it to know that food tastes good when we're hungry, water tastes good when we're thirsty, a fire is fantastic when you're cold, sex is good most of the time. And as a neurobiologist, that I believe that does not happen magically. There are mechanisms of our brain that were very important for our survival that told us this feels good, or conversely, this feels bad. And so the connection here is, whenever it was, in the 90s, however long, I made a very similar, very simple, intuitive biological assumption that when all is said and done, a lot of behavior can be explained by the simple principles that we repeat behaviors that lead to reward, and we avoid behaviors that lead to pain or punishment. And I actually really believe that. It's an old idea, it's intuitively obvious, but it makes a lot of sense. And so I'm digressing a little bit. So in the 90s, both because I was looking for ways of looking for plastic changes, I was interested in that basic principle of behavior. I started working on synapses in the so-called mesolimbic reward circuitry. These were connections onto midbrain dopamine neurons in a part of the brain called the ventral tegmental area, synapses on a major target of these dopamine neurons called the nucleus accumbens, and how they're modified by drugs of abuse. And in fact, that led to a whole field of looking for these synaptic changes, and now, which we'll talk about maybe later in this conversation, trying to use that information therapeutically in ways that we just heard about in some of the talks, which we can get back to. So for a number of years, I was working on how drugs of abuse modify this reward circuitry. And then it's kind of, again, obvious when you start thinking about this concept of reward circuitry and how it can be modified both adaptively and pathologically. It's again kind of intuitively obvious that this circuitry is really important not only for addiction, where drugs of abuse are kind of taking it over. It's also important for depression. So if you think about depression, a core symptom of depression is what we call anhedonia, the inability to experience joy or happiness or reward. And by definition, when you're really depressed, nothing makes you feel good anymore. You're not interested in sex, you're not hungry anymore. And it's almost a circular or tautological argument. If there is a mechanism in the brain telling you something feels good when it's supposed to, that ain't working during depression. So I've done some work in animal models of depression trying to figure that out. So that eventually is leading me to tell you about the work I'm presenting here at this Cold Spring Harbor meeting.
So there's been a number of papers. There's been, the work that you're presenting on serotonin, and then somewhat related to that, maybe very related, is the work that you did on oxytocin.
And I'm leading up to that.
Okay,
so
go ahead.
That's exactly where I'm going.
Okay.
But thank you. So we were thinking about reward circuitry, its role in addiction, its role in depression, and then with some postdocs we were thinking, and again it's kind of, I would almost argue intuitively obvious,
Right.
that social interactions
Right.
are frequently highly rewarding or highly aversive.
Mm-hmm.
And any of us who have been married know that's exactly the case. And in fact, what's remarkable is that same social experience, if you've been married, you know what I'm talking about, the person you're married to can be the love of your life, and 3 minutes later can be the most aversive stimulus you've ever experienced. And I don't believe that happens magically. I believe there are neural mechanisms that explain that. And then understanding how social behaviors can be reinforcing is obviously important for understanding major symptoms of disorders like autism, where one of the cardinal features of autism spectrum disorders is the inability to make social contact. And one possible contributing factor to that is maybe people with autism spectrum disorder don't experience the reward that most people get when they're meeting somebody they really like or hanging out with. It's also relevant, I mean, not to get too political, but I can't imagine a more important topic in today's world, and how fractured our world is, not only in the United States, but worldwide, of if we, I passionately believe if we can figure out the neural mechanisms that allow social interactions to be positive, to be good, if we can figure out the neural mechanisms that allow for compassion and empathy, I actually don't think there's a more important topic in the
So you're
modern
saying
neuroscience
that some
world
of
today.
these mechanisms may even exist en masse at the societal level?
Absolutely. Oh, I think absolutely, because societies are made up of individual humans, and I will get back to oxytocin and serotonin in a second, but what's so incredible about the human species, which is a byproduct, I would argue, of our brains, is our ability of enormous creativity, enormous compassion, enormous empathy, and that this same brain, because of its evolutionary heritage, is capable of unbelievable evil and malevolence and destruction. And I think neuroscience has a lot to say about that. And I literally think we should be spending more effort on the neuromechanisms underlying our most positive human traits. And I believe those can be studied in animals, because I think they're evolutionarily conserved, which gets me back to the question of my presentation here, which is we were thinking about reward circuitry and how it may play a role in social behaviors, in particular what we call positive pro-social behaviors. And as you and the listening audience may know, oxytocin is a neuropeptide that over the last 10 or 20 years has gotten enormous attention. Some people call it the love hormone, which is obviously a gross oversimplification, but it is a neuropeptide that has been evolutionarily conserved, that plays a role in parturition, plays a role in the milk reflex when moms deliver, plays an important role in infant maternal bonding. It's being tested therapeutically as a treatment to enhance positive social interactions. So we looked at the literature. These were not completely original ideas, and we said, well, if oxytocin is really important for certain types of social behaviors, and certain social behaviors are reinforcing, it makes sense that maybe oxytocin can somehow modulate this reward circuitry during non-sexual but pro-social behaviors. So we did my postdocs actually did a body of work where in some published papers, in a paper published in Nature, we reported that oxytocin action in the nucleus accumbens is important for what we call social reward, which is the reinforcing aspect of a social experience. We also showed in a recent paper in Science that oxytocin influences the activity of these VTA dopamine neurons. So nothing is simple, the brain's complicated. It's not just one neuropeptide acting on one brain region. It's a distributed network and modulation of a complex distributed network. This finally gets me to my presentation here. It turns out that the actions of oxytocin in this target region, nucleus accumbens, all of our data forced us to reach a somewhat complicated conclusion, which I didn't like, even though it was published in a high-impact journal, and the conclusion was that oxytocin is working in the nucleus accumbens. The hypothesis was by acting on the terminals of serotonin inputs into the nucleus accumbens and causing the release of serotonin. And that was the concluding hypothesis of that paper. So that body of work immediately led to the next hypothesis. Well, if that's true, if oxytocin, at least in this target structure, the nucleus accumbens, is actually working via influencing serotonin levels, we should be able to go into a mouse brain and manipulate serotonin levels, specifically in the nucleus accumbens, and have very robust effects on social behaviors in our test animals, which are mice. And remarkably, the talk I'm going to give on Saturday during this meeting, and the paper should be accepted soon for publication in a prominent journal, shows some of the most robust behavioral results I have ever seen in my 20 years of doing these sorts of manipulations. So what I will be reporting is that manipulating serotonin release using this methodology called optogenetics, which many neuroscientists know about, the ability to activate or inhibit specific sets of neurons or their axonal projections. If we release serotonin in the nucleus accumbens, we enhance social behaviors. Actually, the term we use is sociability. So we enhance the likelihood that our test animal will, in a non-aggressive way, interact with a little buddy mouse that it's interacting with. If we inhibit serotonin release, we reduce sociability, reduce this social interactions. And then what really made the story more impactful and gets back to my therapeutic interest is we then took a genetically based mouse model of an autism spectrum disorder. This is a chromosomal copy number variation that several investigators here at Cold Spring Harbor are studying. It's called a 16p11.2 deletion syndrome. And we have a mouse model that colleagues at Stanford made where this chromosomal segment is, the term is floxed, which allows us in a temporally and spatially controlled way to go into the mouse brain and genetically delete this chromosomal segment in any population of neurons we want, with, given some constraints. So what we found is if we genetically delete this chromosomal segment that is associated with autism spectrum disorders from serotonin neurons in the mouse brain, the animals show a social behavior deficit. If we record from these serotonin neurons, their activity, which normally increases during a social interaction, is dramatically reduced. And that if we then use this tool of optogenetics to cause the release of serotonin in these, in quotes, autistic mice, we can rescue the deficits in their social behaviors. And that requires release of serotonin in the nucleus accumbens at a specific serotonin receptor called the serotonin 1B receptor.
Your talk is also going to mention some work you've done with MDMA.
Yeah.
That's related to this?
That's related. And the truth is, I didn't realize the talks are only 20 minutes, so I will certainly not have time to get to my MDMA work. And what makes science fun, although I have to be honest, I'm a little almost scared because everything is coming together scientifically so well. So as you may know, MDMA is a drug also called ecstasy that human beings have been taking recreationally for many decades now. And what got me interested in it is that even though it is an amphetamine derivative, which puts it in the class of drugs along with methamphetamine and cocaine, unlike these other psychostimulant drugs, MDMA has qualitatively different effects in human beings, and in that it has this remarkable ability to open up human beings to positive pro-social interactions. Some people call it an empathogen. I'm not sure it's enhancing empathy per se. It matters how you define empathy. But there is no doubt from all the reading I've done, all the anecdotal talk, all the anecdotes I've heard about people on MDMA, I have never read an article or met a person who has taken MDMA who has not had the experience of being more open to a social interaction, wanting to get to know the people in their immediate environment with no aggressive taint to that interaction, only the most positive kind of compassionate, empathic interaction. And I find it remarkable that a drug can reveal this fundamental property of our human brains. And as you know, I've written a commentary in Cell about this, saying this should be the object of scientific study.
So we have to wrap up, but let me ask you, how does the MDMA work relate to what you just talked about with serotonin? And last, do you find it ironic at all that your interest has been in drugs of abuse, but some of the solutions to the problems created by drugs of abuse may actually be solved in some cases in terms of the synaptic plasticity by drugs that are considered illegal now?
Yeah, I think it's a great point. So the reason the MDMA work is related to the serotonin work I talked about is it turns out we have data that the major molecular target of MDMA is something called the serotonin transporter, or SERT. And it turns out when MDMA binds to SERT, in very complicated ways, it causes the massive release of serotonin. And a release of serotonin that is much greater than traditional antidepressants like Prozac, which work via a different mechanism. So it turns out that we have a dose of MDMA that is not reinforcing in and of itself in the mice, which we can show enhances pro-social behaviors in our mice. And we have data that I won't have time to present that we can mimic this effect of MDMA when it's given parenterally, when we directly inject it into the nucleus accumbens. And we can actually block the effect of total brain exposure to MDMA when we genetically delete the serotonin transporter in the nucleus accumbens. So all of this is coming together saying MDMA is kind of the equivalent of our optogenetic release of serotonin in the accumbens. And these were two separate projects that we had no idea whether they were going to come together.
So it's a singularity.
It's a singularity. And then to answer your final question, the lectures I give to medical students and graduate students is drugs are drugs. They don't have evil and good. Every drug that we've ever created, including aspirin and ibuprofen, has good parts to it and bad side effects. And I personally think no drug should be demonized. I think they should be studied ethically and rigorously. And the reason I'm so interested in them is drugs are very useful probes of the nervous system because they have molecular targets.
And there's a lack of good drugs for the nervous system.
And there's a real lack of new drugs for the nervous system. As you know, no real drug with a really novel mechanism of action has been created by the pharmaceutical industry in 50 years. It's all serendipity. And so yes, MDMA is being tested now therapeutically to enhance the therapeutic alliance between a therapist and a patient suffering from PTSD, with preliminary results saying it might have great efficacy. It's being tested in autism spectrum disordered, high-functioning autistic adults. Do I think the reason I'm also interested in understanding its mechanism is it does have some bad stuff. It can cause toxicity if you take too much of it. So I am not advocating everybody should go out and take MDMA. I'm advocating we should study it scientifically, and by understanding its mechanism, maybe we can make an MDMA that has enormous therapeutic efficacy with much reduced side effects, and that would be a great new drug.
So just to finish up, a font of new ideas in the neuroscience field for therapies are very old drugs that were prohibited from being studied, like MDMA, ketamine, and others that are now, and psilocybin, for instance, that are now looking like they may have in the proper setting, some real positive benefits that other existing drugs that you can go buy at the pharmacy really don't.
I think that's a really good summary. And it means, we should be scientists and be allowed to study in a rigorous, ethical manner, and obviously with integrity, watching out for the safety of our animals and our patients. Substances like that have previously been demonized, because giving an animal, and in fact a human being, a drug is no different than running them through a maze or giving them some form of psychotherapy.
Thank you very much.
Yeah, thank you.
Okay.